Hello, I'm Tomas Hanke and I'm from University of Oxford. Our aim is to develop an HIV vaccine which induces effective cytotoxic T cells. And effective really is the important word in the title. Cytotoxic T cells will complement broadly neutralizing antibodies while their induction is suboptimal from prevention of HIV infection. And effective cytotoxic T cells might be key for HIV cure. What seems to be clear that inducing more of the same CTL is not going to work. Natural HIV infection induces strong cytotoxic T cells, and eventually they don't work. They don't prevent development of AIDS. Just reactivating cytotoxic T cells from before antiretroviral treatment is not going to work because HIV has already escaped these cytotoxic T cells. So we just have to do something different. The biggest challenge for development of HIV vaccines is diversity and escape. And that this is the same problem for antibodies and cytotoxic T cells. We're focusing on T cells, and there are various ways how to tackle HIV diversity. And our strategy is to focus the immune responses on the most conserved regions of HIV. This is because these most conserved regions of HIV are common to most global HIV variants, and also mutations in them, if HIV tries to change them and escape, cause replicative fitness loss. So our first generation vaccine, based on conserved regions, showed the principle. The principle that if we take subdominant epitopes from the context of whole proteins or the whole virus, and present them to the immune system using a potent regimen of chimp adenovirus prime and MVA boost, we can induce potent cytotoxic T cells in humans, which can in vitro inhibit HIV replication. And this is the most relevant assay for induction of cytotoxic T cells before we actually start to evaluate their protective efficacy in humans. So our first generation proved the concept, and this poster that I'm presenting here is about the second generation. The second generation has significant improvements over the first generation in several ways. The central idea is the same. We are focusing the immune responses on the most conserved regions of the HIV proteome. But we are enhancing the efficacy of these responses in three different ways. The first way is that instead of consensus clade sequence, we have calculated bivalent mosaic. Bivalent mosaic increases the perfect match to global HIV variants for potential T-cell 9-mer epitopes. And this is shown on the first figure. So the first figure shows the coverage of the bivalent mosaic to global variants in the group M. The more red it is, the better coverage there is. So we are looking for the most red regions, and this is GAG, and this is POL, and this is ENF, and these are the other smaller proteins. So we have selected 6 most conserved regions, 2 in GAG and 4 in POL. Now, if you look here at this figure, this is the global coverage. Again, the more red it is, the better of the second generation. This compares to the first generation. We can see it's less red, and this is what's left in the proteome behind. So we are really selecting the most conserved regions. Now, the second improvement is that we're including protective epitopes, which have now been defined on cohorts on treatment-naive individuals on 3 different continents. they are shown in blue and green little squares here. So we are including the protective epitopes, and finally we are reshuffling these 6 regions in 6 different orders, and this is to minimize the induction of cytotoxic T cells to junctional epitopes. Junctional epitopes are epitopes between 2 regions, 2 adjacent regions, and these are irrelevant because they are not present on HIV. The 2 mosaic 1 and mosaic 2, differ by about 10 amino acids, and they are always used together for immunization. They can be delivered by DNA or MVA or chimp adenovirus. So for each dose we have half the dose of the junction, and we never boost with the same junction again when we use the other vector for prime and boost. So we have 6 different proteins. We have confirmed the induction, the expression of these proteins in human cells. And the following figure now compares the immunogenicity of the 1st generation and the 2nd generation in mice. And the bottom line is that the 1st and 2nd generations are equally immunogenic. And this might seem that it's not good enough, but it is a very good start, because the 1st generation induced the highest frequencies of cytotoxic T cells today in the field, with mean of 5,500 interferon gamma ELISpots per in human volunteer, or human volunteers responded to vaccination. So if we show equivalence to the 1st generation, this is actually a very good start. We have also shown in mice that if we do prime boost combinations, we are inducing strong immune responses, but we are not inducing any responses to pool of peptides which have been designed to cover the junctional epitopes. The last part of the poster demonstrates that we are indeed trying to induce useful immune responses. So we went to a cohort of treatment-naive patients in Japan, and we have tested their immune responses in ELISpots to peptides derived from our immunogen. And we have shown that the higher the magnitude of these responses in this cohort, and the broader the immune responses that these patients have, the lower the virus load and the higher the CD4. So this is the magnitude. This is the higher magnitude, the lower virus load, the higher magnitude, the higher CD4 T cells. This shows the breadth in terms of the number of 10 pools of these peptides that the patients are responding. The broader the response, the lower the virus load, the broader the response, the higher the CD4 cells, the higher the preservation of the immune system. If we take out protective HLA alleles, or patients which have protective alleles, we lose the significance of the correlation between the virus load, but we have maintained the significance of the magnitude and the breadth with the CD4 cell count, and that is the more important clinical observation. So this last figure suggests that we are potentially going to induce protective immune responses that can benefit the patients. So that's where we are at the moment. We are currently manufacturing the GMP vaccines, and we are getting ready for the first small clinical trial in Oxford, and then perhaps bigger clinical trial in around Lake Victoria in Africa. That will be the prophylactic use, and also we have a few therapeutic trials in the pipeline. And that concludes my presentation. Thank you for your time and attention.